Yeah, maybe I should explain why someone who's been a fairly hardcore echinoderm worker for most of their lives, working on Cretaceous, got into barnacles, of all things. And I was doing a sabbatical in Copenhagen a few years ago, and one of my favourite localities in the world is Everklack in southern Sweden, which is a Cretaceous rocky shoreline. So you have boulders with zoned epifauna and all that sort of stuff. And the sediment between the boulders is soft, and it's full of fossils. And it turned out to be full of barnacles. The echinoderms were rather boring, but the barnacles were fantastic because this was a time, late Cretaceous, when the balanomorphs, those dominant acorn barnacles of the intertidal, were just beginning to evolve, and it actually yielded a story demonstrating the existence of the basal balanomorphs and how they originated, which I published this year in Journal of Systematic Palaeontology. But today I'm going to look at another. So while I was on barnacles, I thought I might as well do the rest of them. And there's this amazing family called the scalpelids, which are deep sea. They have over 250 species identified, very distinctive monophyletic group. I'll show you one in a minute. They have a fantastic fossil record. They originally were in shallow water. They've disappeared from shallow water now, and they only occur in the deep sea. And the thing that fascinates zoologists is their reproductive biology, because the deep sea ones have developed these things that Darwin called dwarf males, which I'll explain about later, which live in cavities within the shell of the female. And as with many groups like this, there's been no real phylogenetic approach to classification. It's been based on characters of the shells without thinking about outgroups or the direction in which evolution has taken. So what do they look like? Well, this is a typical nice little scalpelid. The valves are made of calcite. They have 14 valves. This is the peduncle here, which will attach to a substrate of some sort, hard substrate, and this end up here opens up to allow the feeding arms out. Darwin named the bit, and we stayed with Darwin's nomenclature ever since the first publication, 1851, in which he described all these morphological. Well, of course, molecular phylogeny is with us, and when my good friend in Copenhagen, Jens Huig, and Benny Chan in Taiwan did a molecular study using a whole suite of mitochondrial and nuclear genes, I think you notice you see in green you have that genus, which is slightly worrying, because it suggests it's massively polyphyletic. And in fact, if you start looking at any of these other genera, we have a Litoscalpellum down here, and we have another Litoscalpellum up there. So, but of course, zoologists these days don't do whole animal zoology, and they certainly don't do hard parts, most of them anyway. So I was asked to look at these organisms and see if I could make any sense of the morphology using the plates. Now I should point out, of course, that these things are arthropods. The arthropodal structures which sit inside the shell, these segmented limbs, the mouth parts and the penis, can of course be studied in modern barnacles. I just didn't do it because it's a fairly serious undertaking to describe the limbs. So I just worked on the plates for the moment, but it's a provisional study and we'll add this eventually. Well, the shells are magnificent, made of calcite, loads of characters, muscle scars. These are the sites for little males to live in. This is a female shell. And by identifying homologies, color-coding homologies, it's possible, I built up a character list of about 62 shell characters for this group, which I thought would probably give me some idea of what was going on using about 15 species, you just use living species for the study. And it's not rocket science to realise that in fact this group splits into two. Big ones, which are hairy, except I've taken the hairs off with bleach, and little ones that are not hairy. But that seems to have been ignored in most of the taxonomic studies. Features like that obviously don't really matter, or were not considered to matter very much. But once you start taking them apart and looking at the plates, the differences become really obvious between these two groups. So these two here, this is a plate called the rostrum. You don't have me to explain to you that they are very different in shape. And one group, the big group, is consistently like this, and the small group is consistently like that. So how do we determine polarity? This is the big deal, because no one had even thought about polarity. Well, we have a nice Cretaceous outgroup, a basal scalpellum barnacle called Cretaescalpellum, of which we have a few beautifully preserved whole individuals and thousands of plates, which allow us to make really detailed comparisons and to work out the polarity of evolution within the crown group, which are the scalpellars themselves. Just to illustrate what I did was basically take each of these plate groups apart, and I think you can also see that there is a progression going up here. This is close to the outgroup, and these go up through, and the more derived ones all have the umbo, the point at which growth commenced, rather low on the outside, and the primitive ones have it apically. There are a whole series of other characters which, like the shape of this plate, changes quite significantly as you go through the sequence of forms. So, I ended up with a very well-supported consensus tree, of which the topology is similar to the molecular tree, but not identical. Essentially, here we have this well-defined group B. These are the small, smooth ones, and these are the big, hairy ones. This group does not fall into the two relatively well-defined groups defined by molecular basis. Rather, it forms a sort of ladder in which characters are added progressively crownward as you go through the group. The other nice thing is, because we're using plates, we can add fossils, so we can say that the origin of the group was greater than 125 million years, and the most penultimately, the derived clade within the crown group here that developed by 78 million years ago, because these things are very common in sediments. These are from the Cretaceous chalk. This is a fossil one, and this is the equivalent plate of a modern one. So you can make very detailed comparisons that permit you to identify clades in the fossil record, which is rather nice. Also, occasionally, we have examples of evolution taking place. This is a very derived modern barnacle called Scalpellum, which has these horn-like carina lata and a number of other features, and these are a series of scalpellids from the Eocene of the Paris Basin, going through from a less derived form through to a more derived form, which seems to record the evolution of Scalpellum, of this highly derived modern taxon, which is extremely successful. It's the one shallow species that actually makes it into shallow water. So did they evolve in the Cretaceous, or, it's a good question, almost all deep sea morphological genera present by the late Cretaceous. So did they actually evolve in these late Cretaceous chalks? Or are they groups which are more ancient, which simply extended onto the shelves when oceanic fascia spread? Well, the other interesting thing is that although we can make a sensible phylogeny, there are consistent differences between Cretaceous taxa and modern ones, irrespective of which clade they belong to. Now, modern ones tend to have a space along here just covered in skin between this plate, the carina, and the turn. But the Cretaceous ones, it's always infilled with calcite. Almost certainly because you don't leave spaces in your shell if there's high predation pressure. Whereas if you live on the abyssal plain, chances of anyone finding you are relatively remote. So I think we can sort out characters which are deep phyletic characters from those which are related to specific life habits. Now, probably the most famous thing about barnacles from a zoological point of view, I need to get the lower part of the screen. Oh, there it is, that's it. Is their reproduction. Barnacles are hermaphrodites, primitively. These ones in a rock pool are filter feeding and reproducing at the same time. This is the penis here. And for animals living so closely together on a rock surface, reproduction is not a problem. You don't exactly need a dating agency. You have your next door neighbour. But for other barnacles, it's much, what's going on there? My God. For other barnacles, right, enough. You have a real problem finding mates. If you are living attached, say, either on the deep sea floor or attached to a substrate with a very limited opportunity, and these things, this Scalpellum, the one I showed you, lives in small colonies. They're hermaphrodites, and they can just about make it with each other. They retain a penis, they're able to cross-fertilise if you're living in that sort of situation. The penis is rather low. However, isolated hermaphrodites can't reproduce. If you're stuck there on a hydroid or whatever, there's not much chance of meeting up with another barnacle. And 25% of the species are actually single. They don't have anyone nearby with which to make little barnacles. And Darwin, when he dissected barnacles, came upon the most remarkable thing. However, the other day I got the curious case in which the female had two little pockets, in each of which she kept a little husband. And here is the little husband. Not much of a life for them. They are reduced to a sac, which contains a testis and produces sperm. And they live in a tiny little cavity on the edge of the female scrotum. And this actually occurs in a number of other deep sea organisms. Quite commonly, it's known as androdoisi. And it's a feature adopted by organisms where there's a low density of individuals and interbreeding is difficult. So there's a strategy by which some of the individual larvae turn into dwarf males. So that's what they do. They sit on the edge of there they are, and there's a cluster of them living. Now, the nice thing is, and this is new, I found this out, that the pits in which the dwarf males live leave a hole in the shell. The female creates a hole in the shell to accommodate the dwarf males. And you can see this is Scalpellum. It's a very distinctive hole. This is a Cretaceous thing called Scalpellum, and this just has this general pit up here. Whereas the mainstream of Scalpellids have this big Y-shaped groove, presumably to accommodate rather a lot of juvenile males. So we can say that this Androdoisi evolved at least 3 times within the barnacles, and it first evolved in the Cretaceous. It was lost. This lineage never made it through the Cretaceous. This lineage evolved it later, post-Cretaceous and Eocene, and this lineage evolved it more recently. So, morphological phylogeny parallels the molecular tree, at least broadly. So we need a bonfire to get rid of the 29 genera, or a large number of them, into which these things have split because they're polyphyletic and paraphyletic. And this very special sexual characteristic of dwarf males evolved at least 3 times, probably more. So we're actually able, from the paleontological viewpoint, to contribute to the understanding of barnacles, which are such a major group for zoologists studying living marine faunas. Thank you.